Well, hi, everyone. I'm Jim Burns. I'm the CEO of Locanabio, and I'd like to start off just by thanking ARM for the opportunity to tell you the story about Locanabio. And as you can probably tell a little bit from this slide, correct the message, change a life, which is our motto. We're about a new genetic medicine platform, which is targeting messenger RNA, dysfunctional disease-causing messenger RNA, in order to change the life of patients with certain types of genetic diseases. And we're doing this in a way which is, I'm going to have to start all
There
over
you
again.
go. That's okay.
I have this, what did I do with it? Oh, here it is. And that was pretty good.
Yeah, so far so good.
I have,
What's funny? We've gotten good at sort of figuring out when it starts to go downhill.
well, what I have a remote changer, a slide changer, and I forgot to plug it in.
Yeah.
All right, so we'll start again. Well, hi, everyone. I'm Jim Burns. I'm the CEO of Locanabio, and as you can probably tell from
Jim, 1 second. Let me. I hadn't muted myself, and I think I might have made some noise. So let me. I'm gonna mute myself again, and then give yourself a countdown and go.
Hi, everyone. I'm Jim Burns. I'm the CEO of Locanabio, and I'd like to start off just by thanking ARM for the opportunity to talk to you today and tell you our story. And as you can probably tell from this slide, we're about correcting the message of dysfunctional RNA that can cause disease and hopefully change the life of patients who have genetic mutations that can benefit from this approach. What's different about our approach? Gosh darn it. Sorry about that.
That's okay.
Okay, we're going to do it one last time, and this time it's going to work.
Third time's a charm. You have a bunch of investors in the room, and they're looking at you.
Yeah, absolutely.
All right, I'm on mute.
Hi, everyone. I'm Jim Burns. I'm the CEO of Locanabio, and I'd like to start off by thanking ARM for the opportunity to tell you our story. And as you can probably tell from this slide, we're about a new genetic medicine, which is intended to correct the message of dysfunctional messenger RNA in order to change the lives of patients with genetic diseases that can benefit from this approach. And our particular approach is very different than other methods of targeting RNA, which are great ways of trying to target disease-causing RNA. Ours is different, though, and will allow certain advantages and features which we think can benefit patients. And what we're doing is combining the power of RNA binding proteins that already exist and deliver these RNA binding proteins by gene therapy. We can also modulate the RNA binding proteins. We can connect them with effector domains that can modify RNA in a number of different ways, which are shown on the wheel on the right. We can destroy RNA, especially repeat expansion RNAs. We can affect splicing, translational enhancement. We can do RNA editing. And because some of our systems are so small, we can actually destroy RNA, and then in the same system, AAV actually replace with a normal gene. So we have RNA binding proteins that target dysfunctional disease-causing RNA. These are very potent systems, very selective. So they're allele selective, so we can ensure that they target the dysfunctional RNA as opposed to wild type. And it targets DNA and not RNA. And I think what's really important about this approach also then, as I was saying, is that we use gene therapy for one-time administration. And recently, you may have seen this, we raised in a Series B, is closed mid-December, $100 million, which gives us a very nice runway for the next few years, which allows us to build out the next phase of the organization, which includes manufacturing as well as clinical, medical, regulatory affairs. So obviously, we'll be investing more and more in development as we advance our programs towards IND-enabling studies and into the clinic. So let me tell you a little bit about the RNA binding protein systems. So at this point, we have 3 systems, and you'll actually see some data on each of these. Most of the data that you'll see is what's in the blue box to the right, which we call our Cas9 systems. So these are CRISPR-Cas9 systems that have been mutated and changed so that they're selective to RNA. We also have two other systems. The one in the middle is called the PUF systems. I'll tell you a little bit about that, as well as Cas13D, and which is a bacterial-based system that's specific for RNA. So let me tell you a little bit about the Cas13D system. So this is a programmable RNA targeting CRISPR system that's specific for RNA, does not target DNA. And it has an endogenous cleavage domains, RNA-specific cleavage domains for HepN. These are relatively small. They're about 3 kilobases. They target specific RNA sequences that we can program with guide RNAs. The PUF systems are a little bit different. These are human-derived systems. Eukaryotes have PUF proteins that are involved in development. They target RNAs during the development phase and modify their translation. And we use what's called the PUM1 protein, which is a human-based PUM protein for our systems. These are relatively small. They're about 1 kilobases. And there's a homology domain within these PUM and PUF proteins that we can program specific to bind to specific RNA bases. So it's a very nice system. It has no nuclease activity, of course, and we add that with our own nucleases that we have intellectual property on. So that's a PUF endonuclease system that we can use for various destruction applications. So we could be working on a lot of things, but the ones that we are working on are at this point are shown here. You'll hear a little bit about our application for this technology platform for myotonic dystrophy, where we target the CUG repeat that causes that disease in the myotonic dystrophy protein kinase gene. We're also focused in neurodegeneration, again, on repeat expansion diseases, which includes Huntington's, SCA1, ALS, the C9orf72 form of ALS, as well as FTD, and then finally in ophthalmology. So I'm going to give you a little bit of flavor of what we're doing in a couple of these diseases. And as you can see, if you look all the way to the right, you can see that we have worldwide rights to all these programs at this point. So let me tell you a little bit about our first, actually the first disease that we went after as a company, which allowed us actually to raise our Series A, which is in myotonic dystrophy. So this is a multisystemic neuromuscular disease. As I mentioned before, it's a mutation in the DMPK gene. And what happens here is that we have a repeat expansion of CUGs or CTGs in the DNA, which gets transcribed into CUG repeats in the 3' UTR portion of this gene. So it's in a non-coding part of the DMPK gene. And once these repeats get to beyond a certain point, let's say 40 or 50 repeats, then they become toxic. And in some individuals, I mean, these can get into the thousands in terms of repeats. And what these repeat expansions are in the RNA do is that they sequester important RNA splicing proteins. And this has a number of effects, including causing the myotonia and cognitive defects, as well as muscle wasting that happens in these patients. It's a fairly large rare disease, about 40,000 patients in the US, and there are no approved therapies. So what we do with our systems is we can design them so they target the repeat expansions themselves. They do not target the coding part of the gene. So they only hit this is an autosomal dominant disease, so we only hit the wild type disease. We leave, or I'm sorry, the disease-causing allele. We do not hit the normal allele. And as I mentioned before, there's no DNA alterations, one-time administration, because it's we package this in an AAV. So let me show you some data that came out last year in September in Nature Biomedical Engineering. And in this system where you're using RCas9, so it was the original system that we used at the very beginning of Locanabio, which is mutated Cas9. So it's it's specific to RNA, and we use a guide specific to the CUG repeats. And you can see in the RNA fish on the left, on the upper left, the toxic RNA. And you can see that once it's been targeted with a system that targets the CUG, that those toxic RNA are destroyed. And importantly, on the bottom two pictures here, you can see the absence of a chloride channel staining for the tissue that has been targeted with the non-targetable CUG, RCAS9, so no effect on the toxic RNA, and where we were able to destroy in the nucleus the toxic RNA, you see a reestablishment of normal chloride channel staining. Also importantly, if you look over on the right, that we looked at about 350 different splicing proteins and saw a return to normal or partial reversal in over 80% of those 350 splicing biomarkers that we looked at. And then, of course, if you're affecting chloride channels, you would expect that you would have some sort of measurable effect on myotonia, which is one of the hallmarks of DM1. And that is indeed the case. On the left, we see IV injection of our system into our animal model. It's called the HSA long repeat model, which has a long repeat of these CUG repeats. And we can see an improvement in myotonia. And then in adult animals on the right, you can also see an improvement in myotonia. So, and this is with our RCAS9 system, but also a system using Cas13D. So it really shows nice progression from the biopsy of the tissue in terms of improvement in eliminating these toxic repeats, and then replenishment of the splicing biomarkers and an improvement in myotonia. Let me give you a couple of examples in the neurodegeneration space. I'm going to start with Huntington's disease. And this, again, is a repeat expansion disease. There are, I think, probably at least 20 CAG repeat expansion diseases. And the one that's more prominent is for Huntington's disease. So this is a CAG repeat expansion. It's actually in the coding section of the Huntington's gene. And this causes, once it gets beyond a certain number of repeats, degeneration of striatal neurons, leading to uncontrolled movements, emotional problems, and dementia. And as I said, this is a fatal disease. There's no treatment for this disease at this time. And it's a large rare disease. Over 40,000 patients are estimated in the US. So our approach specifically targets the toxic CAG repeats, leaves the normal gene alone, which I think is very important. There's no DNA alteration, one-time administration again, because it's we're delivering this with AAV. One of the things to also that is nice about this approach is that because we're targeting the CAG repeat, we can apply this to other genes that have CAG repeat, such as ataxin-1, where the CAG repeats in that gene cause SCA1. So once we have a formulation that we're taking forward for Huntington's, we have a formulation that we can take forward for many applications. So let me just show you a little bit of our data. This is in vitro and a cell culture test system. And you can see for both our Cas13D systems, as well as our PUF system and PUF E17, the E17 refers to our proprietary nuclease that we marry up with the PUF. We can see that we have a reduction, a knockdown in the toxic RNA. And each of these bars is a different guide that we use for the Cas13D or a different construct for the PUF E17. And then you also see on the right, so the CAG is toxic in of itself, these are RNAs, but also these can form toxic proteins, polyglutamine tracks, and you can see on the right the elimination of those tracks with treatment in these cells. So what we're doing now is looking at our Cas13D and PUF systems in vivo in two different animal models to then make a choice on which one we would go forward with. Just like to show you a little bit of in vivo data with the PUF system, because this is really the first time that PUF systems have been put into animals. And this is with a striatal delivery, again, thinking about CAG repeat disease, well, the specific CAG repeat disease, Huntington's. And what we can see is a very nice expression of this PUF E17 system in the striatum after injection. And this is out to 3 months. So very nice sustained expression of the system. The last system I want to talk about, or disease, is another repeat expansion disease for ALS. So it's a mutation in the C9orf72 gene. Again, it's a repeat expansion of a hexanucleotide, in this case, of G4C2, which is the sense, as well as the antisense C4G2. And what's interesting about this is both the sense transcript and the antisense are both toxic. So to have an effective therapy for C9orf72, it'd be optimum to have a system, a single system that can attack both the sense and the antisense, which we think we're going to be able to do. So this is the most common familial form of C9orf72, and there are about 1,600 patients in the US. The same mutation also causes Ph.D. So let me just show you real quick a little bit of that, of some of our data. And on the left, you see the data that shows knockdown of the sense repeat, G4C2. And if you look in the middle section here, you see one of our favorite Cas13Ds, we call it L4. And you can see that with either of the guides that we have there, over 90% knockdown of the sense. And then on the right, you can see, again, in the middle section for the Cas13D L4 with two different guides, we see a pretty good knockdown, about 75% for the antisense. And so what's important about this is because these systems are small, they're about, Cas13D is about 3 kilobases. We can put the Cas13D as well as both guides in the same system for treatment. So you have a single product for treating the sense as well as the antisense. So we have a busy pipeline. You have a good sense of the progress that we're making already. We have plans for the beginning part of this year, the first half of the year, that we'll be selecting candidates for moving into IND enabling studies in DM1, as well as the CAG repeat diseases that I mentioned, in particular for Huntington's disease, as well as a little bit later in the year for C9orf72. I didn't talk about our retinal programs. I would love to do that, and we can do that another time. So I'd like to thank you for your attention, and I hope to talk to you again soon in the future about the in vivo data that we're going to have in some of our new programs, as well as what we're doing in retinal disease. So thank you.